Hello, my name is Arinze Awagu, and today I'll be discussing our research on how CEP4A depletion affects mergy localization in Mycobacterium smegmatis in cell division. So bacterial cell division is a complex process requiring precise coordination of cell wall enzymes and regulatory proteins at the mid-cell division site. However, in mycobacteria, the exact mechanism by which these enzymes, are recruited remains unclear. And there's certain species like E. coli, which we have a relatively clear picture of how they do this. But in mycobacteria, that's still something that is an area that has potential for more studies. And so one enzyme that plays a role in mycobacterial cell division is going to be MERG. MERG plays a crucial role in the biosynthesis of peptidoglycan. Peptidoglycan is the structural component of bacterial cell walls. And specifically, what MERG does is that it catalyzes the conversion of lipid 1 into lipid 2. And lipid 2 is a key precursor for peptidoglycan synthesis. On the other hand, uh, we have CEP4A, which is an essential protein for mycobacterial cell division. Previous studies have shown that depleting CEP4A leads to continued elongation and defective division, suggesting that it plays a regulatory role. So our study aimed to determine whether CEP4A is necessary for the proper localization of mergy during cell division. So to investigate this, we use a strain of Mycobacterium smegmatis in which CEP4A depletion can be induced. This was achieved by the addition of a DAS tag, which will be recognized by SSPP and tagged for degradation by CLP protein, which is a protease. Um, we also did a dependency localization experiment, which involved protein, fluorescent protein fusion of MERG. So MERG was fused with RFP, red fluorescent protein, and its localization and distribution was tracked using fluorescent microscopy upon CEP4A depletion. So our results show that when CEP4A is depleted, as shown in red, MERG becomes mislocalized. This suggests that CEP4A plays a role in recruiting MERG to the mid cell. And as we can see here in the blue is the cells that were not depleted with CEP4A, and we can see that the MERG remains localized at the mid-cell division site. This finding highlights CEP4A as a potential regulator for cytokinesis in mycobacteria, although the exact mechanism remains uncertain. So in conclusion, our study provides insights into the role of CEP4A in bacterial cell division, and understanding this relationship not only expands our knowledge of mycobacterial cell biology, but could also contribute to future therapeutic strategies targeting bacterial cell growth and division, especially important for pathogenic species like Mycobacterium tuberculosis, which is the causative agent of tuberculosis and still remains one very difficult infection to treat. Thank you for your time, and I look forward to your questions and hearing back from you.